Colonizer, Android OS based automated microbial colony counter. Abstract: colony counting to estimate colony forming units (CFUs) is a standard microbiology procedure used for determining the number of viable microbes in a system. Typically, this process is done manually, which is time-consuming, tedious, and sometimes error-prone. Alternate image analysis-based systems are also time-consuming. In this paper, we develop and demonstrate a simple Android-based automated colony counter, called Colonizer, also made freely available on Google Playstore. Our algorithm can also threshold and segment overlapping colonies and thus provide rapid, accurate counts across a wide range of colony densities, from 50 to 500 colony plates, for different microbes using any Android-based phone or device. Introduction: CFUs, colony-forming units, are estimates of the numbers of viable microbes, bacteria, archaea, or fungi in a given system, and has been microbiology staple for decades. Counting of CFUs provides information on how microbial cell viability is affected by different environmental or pharmacological conditions. Typically, the estimation is performed by planting microbial cultures on nutrient agar, petri dish, and counting the colonies formed manually. This is a time-consuming, tedious, and error-prone process widely despised by most student researchers, and the tedium and error due to manual CFU estimation has not changed. Efforts to automate the estimation of CFUs include using a DSLR camera or a flatbed scanner from image acquisition connected to a computer. For example, Putnam et al. In 2005 previously suggested a setup with a Canon PowerShot A75 coupled with a commercial Protocol 3 counter for automated colony counting. Shang et al. In 2009 designed an automatic bacterial colony counting system using a Nikon D50 DSLR for image acquisition and an Intel Core 2 Duo PC for running the watershed algorithm for cementing colonies. Recently, Brugger et al. In 2012 came up with a hardware setup for automatic counting of bacterial CFUs on agar plates. They use a circular dark field illuminator with an UAUY UE 1640C camera for imaging and PC for adaptive thresholding and cementing colonies. These setups require additional hardware components, some training or learning, and technical expertise for the design of the system. However, with a viable processing power present in typical smartphones, most image processing algorithms can be run on the smartphone itself. Hence, we develop a mobile Android-based software, Colonizer, combining the image acquisition and analysis steps inside the smartphone. We also suggest a simple setup to ensure uniform lighting conditions for better accuracy. Using this, no manual selection of region of interest or tweaking of parameters is required. Simply taking the picture of the petri dish is sufficient. The end result is inaccessible, portable, and accurate colony counting system for estimating CFUs across a range of colony densities. 500 colony plates for typical laboratory microbes such as E. coli and S. cerevisiae. Results and discussion. Before we describe the algorithm developed, we first recommend a way to take petri plate images so as to minimize errors due to poor lighting or image quality. We use a small cardboard box, as shown in Figure 1AB, with a small hole to ensure uniform illumination, as described in the materials on metals. With setups, this setup reduces interference due to non-uniform lighting and removes any noise from the edges of petri dish. These setups take 5 minutes to build with readily available components and avoid noise due to gray shiny colonies, foam shadows, etc., which can appear in the petri plate is imaged directly in ambient lighting. We also provided a schematic illustration of the construction, Figure 1C. Apart from the cardboard box setup, we also tested the Android app in a standard laminar flow hood and under viable ambient lighting conditions. The petri dish was kept vertically in the hood to ensure uniform illumination and to avoid reflections and glare. Analysis of the data for all three methods described sequentially suggests that images with unwanted light reflections or poor quality will reduce accuracy as shown in Figure 2AB, etc. when the images are clicked outside the hood under ambient light conditions. Algorithm overview. The image processing algorithm was written using standard OpenCV library function. The first step of this algorithm is to apply to a top-hat transformation of the selected image to correct non-uniform illumination. Since non-uniform illumination can result in noise and artifacts leading to false counts, it is important to maintain uniformity in illumination for the image. Top-down transform achieves this by adopting a background equalization which helps in features extraction in an image. The image is then binarized through Otsu there's holding 9, 10, 11. The algorithm then detects all filters out and abnormally sized contours and draws the remaining ones on a black background. Following this, a distance transform for the black and the white contours image is performed. This step is necessary to segment connected contours into individual components. By finding the regional maximum of the distance transform contours can be estimated centers of the colonies. Thus, count gives the numbers of colonies. These processes are illustrated in Figure 3 and 4. Thus, through these methods, the algorithm can count colonies which are connected to each other of overlapping. Execution of the different steps of the algorithm finally counts the number of colonies in the plate which is displayed on the screen below the selected image. Algorithm features. A. Tophat transform. The intensity values in a grayscale image can be considered as elevations such that the image is composed of mountain tops, bright colonies, and valley lows, dark background. Uneven illumination, which leads to uneven contrast in an image, often degrades the threshold separation of adjacent mountain peaks between a valley. To correct this and procedure an image with enhanced contrast, the two-phase transformation is applied on an image. Figure 4 and Figure 4a illustrate how the two-phase transform works. B. Otsus thresholding. Otsus thresholding is a binarization algorithm used in image processing. 9. It involves iterating through all possible threshold values and classifying each pixel as either background or foreground depending on whether it is larger than the threshold. It's larger than the threshold. The algorithm finds the optimum threshold where the inter-class variance between foreground and the background pixels are maximized. This helps producing an image with a sharp contrast difference between the background and foreground, and hence make it possible to detect binary contours in the image. Outlier and noise removal. The steps involves removing artifacts and noise present in the images by detecting counters. We remove potentially noisy counters by filtering out the ones having an area less than 12 pixel units, applying distance transform and finding the regional maxima. The distance transform is an operator applied to binary images. It transforms the input image into a gray level image where the gray level intensities of points inside foreground regions are changed to show the distance to closest boundary for each point. Following this, the regional maximum of distance transformed image inserted out. This helps to separate overlapping contours in the petri dish images and thereby ensures that each colony is a cluster is counted as individual. Accuracy. The accuracy for Android app was determined by comparing the manual counts with the algorithm count values. S. cerevisiae and E. coli images were separately analyzed to determine the accuracy of designed algorithms. The accuracy was subject to the image acquisition method used. A mean accuracy, respectively, was observed for images taken within a laminar hood, images taken using a cardboard box setup, and images taken to bench ambiental light respectively. Image taken under ambient lighting shows substantially lower accuracy and higher fluctuation than the box and the hood, and also illustrated in Figure A. We also tested the app performance across different devices and found low variation among them. Thus, we conclude that the most accurate count with a low variance is obtained by taking image inside the hood or by using the cardboard box, and both of these methods are significantly better than taking images in ambient conditions. Repeatability. To ensure that the app performs consistently, we took multiple measurements of the same plate on 2 different phones. Low variance count on both shows that the algorithm counts do not fluctuate on repeating the measurement. Table 1 illustrates the entire dataset of counting colonies by using multiple smartphone models under different image acquisition conditions. Speed. The speed of the application depends upon the processing capacity of the phone used. We tested the algorithm on the smartphones with both very low end Micromax Key Q4014 and a higher end processor Moto G4 Plus. The execution time ranges from 20 seconds for the Micromax Key 4014 to 8 seconds for 20 seconds for 8 seconds for Moto G4 Plus. It is to be noted that the Colonizer app does not require the user to manually select any parameters, and the image processing algorithm is completely automated. Thus, it can be stated that significantly short time span is required to analyze the image and produce the count. We conclude by summarizing that Colonizer is an easily usable, accurate application which operates on any Android OS based device for estimating CFUs in a range of typical laboratory microbes, from bacteria to yeasts. The application is free for download and use on Google Playstore, and the source of code is open. We hope the application is widely adopted by students and microbiological research. Material and methods. Strains and grow conditions. Cerevisiae colony grow yeast colonies grow on yeast extract peptone dextrose EYPD agar in standard 9 cm petri dishes and grow at 30 Celsius were used to collect sample images. A single batch of yeast colonies of varying densities between 550 and 650 were grown on 4 agar plates. E. coli colony grow bacterial colony grown on LB agar in standard 9 cm petri dishes were used as sample images. Bacterial colonies of slightly varying densities of range of 50-400 were grown on agar plates using standard microbiological procedure and grown at 37 Celsius. Card box set up for correct lighting. A small cardboard box illustrated in figure 2 with a movable lid and small hole was used to ensure uniform illumination. One face of the box is kept open to ambient light, which acts as the light resource for imaging. The petri dish sits at the bottom of the box on a black sheet. The smartphone is attached to the lid such that the camera lens aligns with the hole in the lid. The lid is vertically adjusted to focus the 8 cropping circle on inside boundary of the petri dish, as also shown in the figure 1D. This removes any noise from the edges of the petri dish. The advantage of this setup is that it could be built with readily available components to avoid noise due to glare, shiny colonies, foam shadow, etc., which can appear if the petri plate is imaged directly in ambient lighting. Testing on different phones. we tested the app on 5 different smartphones running Android OS with Micromax Key 14, Lenovo Vibe, HTC, Moto G, and Moto G4 Plus. The camera quality ranged from 5 to 16 megapixels, and all with built-in CMOS sensors. Acknowledgements. The author thanks Snidaha Das Vinedvi Shina Radisha Visomia Pabitro Nandi and Lashia Samhita for providing us with microbiology microbial plates, smartphones app, and apps validation and testing. SL is founded through a Wellcome Trust DBT EA intermediate fellowships